Sept there. Yes, sir.
So before we get started, one quick announcement. Next week, Pete Bauman is going to be presenting in seminar. He'll be talking about his new data layer for undisturbed prairie over eastern South Dakota. And then he is also going to be doing a tutorial on that data layer after class. He'll be able to spend until about 12:30 or 12:45 showing people how to use his new data layer, and we're going to be doing that over in Wecoda. I believe the room number is 6, but I will send out a reminder about that with additional information. So in that room we will have everybody who wants to attend will be able to use a computer with ArcGIS on it and play with Pete's data layers as he is telling you about them, okay? This week we have the pleasure of having Dr. Eric Michel, who's a postdoc with John Jenks, talking with us today. Eric comes to us from storied wildlife programs at UW-Stevens Point, as well as Mississippi State. He is a postdoc, as I said, with John Jenks. Here he'll be working on survival and movement of adult and fawns of white-tailed deer in the Dakotas. And today he's going to be talking to us about assessing factors that affect individual and population level morphometrics for captive male white-tailed deer. So with that, welcome.
Thanks, Josh. Thank you all for coming out today. I'm always excited when I have an opportunity to discuss some of my previous research. So like Josh said, I'm originally from Wisconsin, go Packers. Went to UW-Stevens Point. From Stevens Point, immediately made the trip down to Mississippi State University. Never really saw myself being in the South for as long as I was, but I was there for almost 7 years doing my master's and my PhD. So I finished up in March and immediately moved back north up here, obviously the SDSU, and it's good to be back at Northern Latitude. So at MSU, during my master's, I did a lot of work assessing different factors that influence rank within a dominance hierarchy for our captive female white-tailed deer at our research unit there. Also looked at whether or not dominance was a beneficial behavior with regards to producing a high-quality offspring. But today I'm going to be talking mostly about my dissertation work. We had a unique opportunity to look at a lot of different data with our captive animals and assess different factors that influence phenotype throughout an individual's life. So we know beyond a doubt that there's 3 main factors that influence phenotype for all mammals, and particularly white-tailed deer. Genetics being that most important factor. That lays the roadwork for what an individual can produce with regards to antler and body size. But with harvested species like deer, genetics is almost impossible to manage in a free-ranging population, so although it's very important, there's not a whole lot we can do about it. Other factors include age. We know that deer aren't going to express their full phenotypic potential until they reach maturity, and then nutrition is another really important factor in that equation. If you don't have high-quality nutrition available, an individual is never going to reach its full phenotypic potential. So we know that these three things are major influences on phenotype. What we don't know as much about is how a mother plays into this equation. If you think about it, gestation for a white-tailed deer is about 6 months long, an additional 3 months during lactation, which is the most energetically expensive period for a mother. So a mother potentially has the opportunity to influence the phenotype of her offspring. And furthermore, if those influences persist throughout time, that could have some implications for inclusive fitness. So like I said, we had a really unique opportunity where we knew who these fawns were being born to within our captive facility, and we were documenting growth throughout 3 years of age for these individuals, so we could start to assess some of these relationships with regards to the mother. So today, we're talking about the 4 objectives I had for my dissertation work. And these 4 objectives are essentially broken down into 3 chapters, with objective 3 and 4 being a single chapter. So first, we wanted to simply assess phenotypic change in response to improved nutrition. That was my first chapter. We'll talk about that shortly here. Second chapter, second objective, essentially I wanted to assess what maternal and early life factors influence offspring phenotype and whether or not those influences persisted throughout life for that offspring. And finally, I wanted to assess the heritability and predictability of antler characteristics. 2 separate objectives, again, condensed into 1 chapter. So we have some real quick general methods that we use to collect data off these individuals. So we'll go through the general methods first, and we'll jump right into my first chapter. Here we're looking at a state of Mississippi, and we collected deer back in 2005 with the help of some of our state biologists from the Mississippi Department of Wildlife, Fisheries, and Parks. And we collected individuals from 3 unique soil regions in the state, our Delta soil region, the fin loss soil region here in the red, and the lower coastal plain, or the LCP soil region in the blue. In this map, each black dot is a capture location. We had several capture locations throughout each soil range to ensure that we had the entire genetic variability within a soil region. And when we brought them back to a research facility, we kept them separated by region. So delta deer were only kept with other delta deer, breeding with other delta deer, and so on and so forth. We allowed them to produce 2 generations of offspring. Our first generation, or F1s, were simply offspring of wild-caught females and raised on a high-quality diet. We were feeding these animals a 20% crude protein pellet from Purina, so that should meet all their nutritional needs. Our second generation, or the F2s, are simply offspring of the F1s. But unlike the F1s, the F2s, not only were they raised on high-quality diet, their mothers were also raised on that same high-quality diet. Each day during the summer, we'd search our pens for fawns. When we found a fawn, we'd give them an ear tag, a unique number, so we could track them throughout their life. We'd take simple morphometric measurements off of them, like body mass, hind foot length, total body length. We'd pull a hair sample for a DNA analysis where we could assign parentage for these fawns. Then we'd take the same morphometric measurements off those fawns once they reached about 6 months of age. We would sedate our adult bucks, 1.5 years and older of age, every fall. Again, taking the same morphometric measurements and also taking antler measurements as well. And finally, we took data off of our females in the fall as well. Again, taking those same morphometric measurements, body mass, hind foot length, and total body length. So moving into my first chapter, again, we just wanted to measure the phenotypic response to improve nutrition for male white-tailed deer. So there's a relatively new branch of science called epigenetics. Essentially, epigenetics describes basically describes how the environment alters the genetic expression of offspring while in utero. There's a lot of evidence that shows that this is occurring in humans. So for example, you have 2 mothers that are pregnant that are observing quite different environments. One mother has a really high quality environment, a lot of high quality nutrition. That's going to influence her offspring's phenotype. The offspring could go on to become a professional quarterback in the NFL, be league MVP, Super Bowl MVP, be quarterback of America's team. But on the other hand, if you have a mother that has a low quality nutritional environment while she's pregnant, she might have the opposite effect. So as an NFC North fan, I think the one thing that we can agree on, that the best thing that's happened to the NFC North is Jay Cutler. He's thrown more interceptions than other teams for touchdowns, I think. That's not really all that surprising, right? The environment that you experience as a mother influences the phenotype of your offspring. But what is surprising is that can then be influencing the grand offspring of that mother. That can be that essentially that environment can be inherited across generations. So the question becomes, if this can occur with humans, does it also occur with white-tailed deer? Can deer inherit their environment? Well, Geis described essentially 2 different types of phenotypes that a population can display in the wild. You essentially have an efficiency phenotype that generally promotes survival over growth of large secondary sexual characteristics like antler and horn size and body mass. Then, if the nutritional environment improves, these individuals potentially can display luxury phenotype, which promotes the growth of large secondary sexual characteristics. So in the wild, we know that generally deer populations are overpopulated, which usually means that their nutritional environment's degraded across time. So pretty safe to assume that in most wild populations, deer are displaying this efficiency phenotype. So the question that we had is then once you improve the nutritional environment, can they switch phenotypes and then display that luxury phenotype? We also know that some morphometrics are going to respond differently to changes in nutrition. So for example, skeletal measurements are going to probably be a little bit less likely to change. They're going to develop relatively well regardless of the nutritional environment. That's because they have some major implications to gaining different resources and survival. For example, in caribou, leg length is related to the ability to traverse snow to get different resources. So longer legs, more efficient they can be in traveling. The common murre, tarsal length is related to ability to escape predators, so directly related to their ability to survive. So again, we'd expect these morphometrics to develop relatively well. So we had a couple of predictions with this chapter. Essentially, the first prediction is we just thought that we were going to see increases in morphometric size in response to improved nutrition. If there's any delayed maternal effects that are occurring from the wild, we're expecting those to be alleviated when we improve the nutrition. Second, we predicted that we're going to see a clear growth hierarchy prioritization with these different metrics. Some metrics are going to respond a little bit quicker than others. So we analyzed these data with Bayesian metrics, with the Markov chain Monte Carlo generalized linear mixed model approach and program R. Essentially, our response variable was going to be those several different morphometrics and their size, as we calculated that with an estimated Boone and Crockett score to encompass the entire size of the antlers, and their mass for weight. We had body mass, total body length, and hind foot length for the response variables. Again, we know that individuals get older, or as they get older, they get bigger, so we included age in the model, included generation in the model, because we wanted to assess whether second generation individuals were bigger than their first generation counterparts. And we included individual ID as a random effect to account for these repeated measurements of individuals across time. Once we had the model output, we then used the MCMC-GLM.predict function to output our population means. So we have all this data, so I'll bring it together. What do we find? Well, first let's look at what variables were significant within our model structure. So we saw that age was significant for all of the response variables. Again, that's not really a surprise. We kind of figured that going into it. We also saw that generation was significant in all variables, for all variables, indicating that second generation individuals were larger than their first generation counterparts. However, region was only significant for the body morphometrics, so body mass, total body length, hind foot length. So this is indicating that there's still regional variation from the three source populations, again, the delta, thin loss, and LCP regions, even after we improve the nutritional environment. So what this looks like graphically, again, these are simple box plots. So we have the predicted antler score and the antler mass for the delta, thin loss, and LCP regions for each generation. These box plots for antler score, this dot dash line is the harvest data. So we have a lot of harvest data in Mississippi, so we're using that for reference for our first generation. The black diamond is the population mean, the gray line is the median, and then the whiskers are mean max and minutes. So you see a general trend across time that individuals are growing larger antlers from first to second generation. But if I can draw your attention to the lower coastal plain population, you see a dramatic increase in antler size from first to second generation. That's about a 75 centimeter improvement or increase from first to second generation. That translates into about 25 inches or so. So I mean, as a hunter, that's a really, really big difference, a really big increase in antler size. So next, let's look at our body morphometrics, the body mass, total body length, and hind foot length. Again, that dashed line is the harvest data we have for body mass. Same general trend, we're seeing increases in body mass and skeletal structures across time. So it's kind of what we expect when you improve the nutrition. But again, for the lower coastal plain deer, you're looking at a really dramatic increase, about 15 kilograms, which again is about 35-pound increase from first to second generation across time. Again, a very, very large difference if you're a hunter.
Eric, what are the sample sizes that you're sharing with us?
Yeah, so they vary by region and generation. We had, our lowest was, I think, 12, 15 individuals for second generation LCPs, and we had 25 individuals for first generation Vinless. So within that range.
Thank you.
Whoop. So we also saw clear growth prioritizations. So once you looked at all the numbers for antler metrics, you saw about a 20% increase in antler size from first to second generation for all three regions, which obviously is a large increase in size. Followed by body mass, about 15% increase, and finally there's only about a 2% increase in the skeletal structures. So we know that once you improve the nutritional environment, antlers are going to show the greatest response, followed by body mass, and finally the skeletal metrics. So with regards to the generational improvement, we saw increases in all morphometrics from first to second generation, suggesting that these individuals can switch their phenotype across time. They can switch from that efficiency to low-grade phenotype, which is really important because individuals are experiencing new environments all the time. We're degrading environments as humans all the time. They're experiencing these new environments. The ability to adapt is allowing them to express new phenotypic optimums, which allows them to persist in that environment. But we also saw generational increases even for deer from our delta solar region. One thing I didn't really talk about is the variation of those soil regions. The Delta is a really high quality soil region, a lot of egg, a lot of high forage production in that region. So we didn't expect deer from that part of the state to really increase antler and body size, but they did. They also had dramatic increases. So we really don't know what the upper limit is for antler and body size for deer in Mississippi. With regards to the growth prioritization hierarchy, generally when you improve the nutritional environment, white-tailed deer are going to invest in the weaponry first, followed by body mass, and then lastly their skeletal metrics. The skeletal structure is highly canalized, meaning that it's going to be pretty well developed across time. The verse for this was actually shown in Canada. And on Anticosti Island, Cote and all, they basically showed that after 25, 30 years of severe habitat degradation, skeletal structure is about the same from beginning of that time period to the end of it for deer up there. So it kind of works in both venues there. We also hypothesized that we saw an epigenetic effect. Essentially, we saw an increase across 2 generations. We don't know, we didn't see any stabilization, so we don't know if that would have continued to increase across 3 generations. Epigenetic effects are the likely mechanisms for maternal effects. So essentially the ability for a mother to communicate the environment that she's experiencing with her offspring while in utero is really, really important, so that phenotype of the offspring matches the environment that it has. It matches the nutritional quality that's about to experience. And this is a much better explanation for heritable variation that we can't explain by DNA structure. Subpopulations, they may be different with their phenotypes, but we don't know if they're actually genetically different. So moving into my second chapter, excuse me. Here we wanted to look at how maternal characteristics and early life characteristics essentially influence mother's offspring and whether or not those influences persisted throughout the offspring's life. So if we're starting off with fawns, there's a lot of different factors that can influence fawn quality. So we know that maternal phenotype, age, and body mass. Generally, heavier mothers and older mothers produce heavier fawns. We know that influences fawn body mass. Birth date. Sometimes some studies have shown that early-born fawns will be heavier than late-born fawns. Previous year reproductive success of the mother. Generally, reproductive success in the previous year can influence the ability for a mother to produce a heavy offspring in the current year. So all these different factors we know influence fawn body mass. But do those influences persist then to juvenile phenotype? Do they persist? Is that offspring generally heavier or lighter due to these influences as a juvenile? Furthermore, does that then influence the adult phenotype? Are these individuals able to compensate, or do experiences that they have early in life persist throughout their life, influencing and their body size later? Well, adult phenotype is really important. You know, if you're a hunter, generally you're interested in antler score, but from the perspective of these animals, phenotype influences reproduction. So if you are unable to compensate, and you have a poor, low-quality phenotype, that's going to directly influence your ability to reproduce as an adult. Furthermore, relating that back to the mother, that could potentially influence her inclusive fitness across her lifespan if her offspring aren't being reproductively successful. So that's a pretty important topic to understand. Well, this topic is often very, very complicated and complex. A lot of these variables are highly correlated. So for example, maternal age and maternal body mass, like I talked about before, you get bigger as you get older. That really makes it difficult to assess the relative influence of each factor on other relationships. So if you're looking at how maternal age and maternal body mass influences the birth mass of a fawn, the correlation between those variables really makes it difficult to assess if maternal body mass is driving that relationship or is it maternal age. And there's several different relationships in these figures I'm about to talk about that are highly correlated, and we wanted to be able to account for those relationships. So we developed 4 different hypotheses to test specific relationships while we're accounting for the correlated variables. So in this figure here, this is our first hypothesis called the direct maternal characteristics model. And in this figure, you're looking at all these black lines. These are the consistent relationships that we held across our 4 model structure. They're always going to be indicated by the black lines. So once we accounted for those correlated variables, basically the relationships that we were looking at was whether or not maternal characteristics such as maternal age, maternal body mass, the previous year cumulative lactation demand, essentially a measure of reproductive success in the previous year, whether those variables had direct influences on yearling, 2-year-old, 3-year-old adult body mass, and antler size. We also included juvenile body mass in this model and all the other models as well. So our second hypothesis was addressing direct effects of early life characteristics. So again, we have all these correlated variables that we're accounting for in the black lines. But the specific relationships we wanted to address with this hypothesis was whether or not early life characteristic of birth date directly influence adult body mass and antler size. Again, holding juvenile body mass in that model as well. But we recognize that it may not be an either-or situation. It may be a combination of maternal and early life characteristics that influence the phenotype of the mother's offspring. So again, holding all those correlated variables constant, these relationships that we were assessing with this hypothesis, basically direct effects from maternal and early life characteristics on adult body mass and antler size. Finally, it may not be a direct influence of any of those variables. It may only be an indirect influence of birth date, maternal body mass, age, previous year, reproductive success, on their influence on juvenile body mass. And then juvenile body mass may persistently influence phenotype throughout the rest of the animal's life. So this is our indirect effects model. We included juvenile body mass in all models because that's a very important characteristic to fawns. Essentially, if you don't reach a critical body mass before your first winter, particularly at northern latitudes, you're probably not going to survive that first winter. So we kept that variable in each hypothesis. So like I said, this is a really complicated scenario. It's really, really hard to analyze. But there is one method that allowed us to look at it. The Shipley's directional separation analysis, or the Sept analysis, allowed us to kind of parse out some of these relationships. Basically, with our data set, we had repeated measurements of mothers across time. We also know that we had that regional variation that I discussed in the previous chapter that we had to account for. So we were able to include those two variables as random effects with this model framework. We then used the Sept analysis to calculate a C-stat, which basically follows a chi-square distribution and basically informs us of model fit for each model. So we had 4 models. The 4 models I just talked about for yearling, 2 and 3-year-olds. So essentially 12 models altogether that we analyzed separately. So each model could potentially fit our data or not. So that C-stat allows us to assess model fit. But because we can have potentially more than one model that fits the data, we had to rank them. So we use that using AICC calculated according to Shipley in 2013. So once we finally get our one top ranked model, we know what relationships exist, but we don't know the influence of those relationships. So we then use the same model structure, or the mixed model structure that we use to assess model fit, to then calculate the standardized regression coefficients to assess the relative influence between those variables. So we find if it's maternal characteristics, early life characteristics, or a combination of both that really influence offspring phenotype, and do those influences persist across time. Well, our top model for all 3 ages was the direct early life characteristics model. Again, that depicts a relationship, a direct influence of birth date on adult body mass and adult antler size. So for our yearlings, we saw a positive relationship between birth mass and juvenile body mass. And that's not really a surprise to us. We saw a negative relationship between birth date and juvenile body mass. Again, not really a surprise. If you're born later, you have generally less time to grow winter. But then again, saw a positive relationship between juvenile body mass and yearling body size and antler size, which is intuitive as well. Even though this model indicated a direct path from birth date to antler size, it wasn't significant, so that's why it's indicated by a dashed line. But what is most interesting in these models is this direct relationship between birth date and adult body mass. It had a positive influence on yearling body mass. So basically, the later you were born, the heavier you were as a 1-year-old, indicating compensation occurred. For 2-year-olds, we saw increasing importance of birth date on 2-year-old body mass and decreasing importance of juvenile body mass on 2-year-old body mass. And then by the time you reach 3 years of age, there's basically the same level of influence for birth date and juvenile body mass on 3-year-old body mass. Again, this is a positive relationship, so this is indicating that late-born individuals generally compensate their body mass by 3 years of age. But we want to make sure of that, so we plotted out our body masses by age class. Again, these are standardized body masses. We standardize them by soil region and generation, so we can account for that variation with those variables and make a valid comparison across time. So we're seeing that late-born individuals in the black bar here are generally quite lighter as juveniles compared to their early-born counterparts. But then by yearling age class, there's less of a difference there. By 2 years of age, you see a reverse in that trend. By 3 years of age, you see a magnitude difference there for late-born versus early-born. Again, this is a real simplification of what we just talked about. That path analysis is the meat and potatoes of it. But this just supports that we're seeing at least compensation occurring within this population. So we know from other literature, particularly with food restriction studies, that compensations occurs. Generally, individuals are not growing at their maximum growth rates. And there's got to be a reason for that. And we think that reason is probably because growing at a maximum growth rate will reduce your lifespan. So it doesn't make sense to grow as fast as you can if it's going to end up costing you your life earlier in time. But if you need to compensate, increase your phenotypic size, which potentially could influence your reproduction later in life, then compensation might be a viable strategy for you. If you're born late, you're born light, and you're not going to really observe a lot of reproductive opportunities later in life, it doesn't matter if you live a long time. But if you compensate, you're now competitive within your environment and can reproduce, that makes up for that shortened lifespan. But we also identified some other relationships in this model as well. Particularly, I want to talk about the relationship of birth mass and birth weight on juvenile body mass. So we think we found a silver spoon effect with all these relationships. Essentially, you're born early, you're born heavy, which means that you're a heavier juvenile, which means that you're potentially a heavier yearling compared to your late-born counterparts. So this might be a strategy to increase your reproductive success earlier in life. If you're heavier within your cohort as a yearling, you should be able to outcompete your late-born, lighter counterparts in the yearling age class. So really, we think we identified alternative strategies to potentially influence reproductive success within our population. Either you're born late, you're born light, you have to compensate across time. You're generally lighter as a juvenile, lighter as a yearling, but then by 2 and 3, you start compensating. You now are observing more reproductive opportunities. That can increase your reproductive fitness across time. Or you have the silver spoon effect where you're born early, born heavy, and you're heavier early on in life, which may increase your reproductive opportunities early on, but then you plateau out when your late-born counterparts catch up later in life. But compensation may not be a viable strategy across the white-tailed range, particularly at northern latitudes where you have very severe winters, like South Dakota. If you're born late and you don't reach that critical mass to survive winter, you never have the opportunity to compensate later in life. In retrospect, in Mississippi, we have fawns from that lower coastal plain population where mean birth date's August 15th. So in the South, you can be born pretty late and still survive the winter, so they have this opportunity to compensate. But it would also be interesting to assess this relationship at northern latitudes when you have mild winters. What is that threshold of winter severity that allows you to be born as late as possible, still survive the winter, and then do you display compensation in that environment as well? So moving in, wrapping this up, going to the last chapter. Again, in this chapter, I wanted to assess predictability and heritability of several different antler characteristics for white-tailed deer. So heritability is a pretty important metric that oftentimes describes the evolutionary trajectory of a species. Essentially, the more heritable a metric is, the more easily it can be changed. That's really important when you're talking about harvested populations. A population of bighorn sheep, there's been 2 studies now that have been done on this single population that have shown that harvesting has actually altered the evolutionary trajectory of horn size in this population. Essentially, you have younger rams that aren't in the breeding population yet, but are meeting the minimum curl criteria for harvest, and they're being harvested out of the population before they can breed, which obviously does not allow them to spread their genetic variability or genetics to that population. There's also been another study that was done in 2013 that showed that general trends of antler and horn size of several harvested species in North America are on the decline. White-tailed deer was one of those species that was listed. So the question is, can hunting potentially alter the evolutionary trajectory of antler size for white-tailed deer? Heritability is also a really important metric used in modeling sometimes controversial management strategies, such as assessing the efficiency of selective harvest on younger aged animals. Heritability is a metric that we use in that model. Also, when you're trying to simulate the effects of releasing captive raised animals into the wild, heritability is a metric in that model as well. So we need to have an accurate estimate of heritability to accurately model these strategies. But there's contradictions in the two estimates that we currently have for heritability. Williams et al. Did a study in 1994 out of Texas that basically showed that several antler characteristics were highly heritable. Heritable ranges from 0 to 1. 0 means a metric isn't heritable at all. 1 is perfect heritability. But you have different categories within that range. So 0 to 0.2 is lowly heritable. 0.2 to 0.4 is moderately heritable. And greater than 0.4 is highly heritable. With metrics that reach heritability of 0.6 or so or higher being pretty rare in population. So Williams et al., those two basal circumference and antler mass were really heritable metrics. But then Lukafar and Jacobson did a study in '98 that showed those same metrics weren't nearly as heritable. So again, when you're trying to model these different management strategies, this becomes really important to have accurate heritability estimates. Switching gears to predictability. Again, when we talk about predictability, we just are meaning using antler size early in life to predict antler size later in life. But the general thought being that if you have large set of antlers as a yearling, you're going to continue to have larger antlers later in life. If you have smaller antlers, you'll continually display smaller antlers later in life. Predictability, well, we know that yearling antler size can be influenced by different maternal factors, litter size, parturition date. That can potentially influence your ability to predict antler size later in life based off of that yearling set of antlers. It also describes their efficiency for selective harvest. You don't have this situation very often in northern latitudes, but in the southeast, you have really highly productive herds. You have a lot of deer on the landscape, and sometimes harvesting does just isn't enough to get your deer density down. So you also have to harvest bucks out of the population to reduce your deer density. Well, if you have specific management goals and a yearling buck doesn't have the potential to reach that antler size that you're looking for as a landowner later in life, you want to harvest them and remove them off the population because they're eating a lot of food. So the earlier you remove them from the population, the more food that's available to other deer that might reach your management goals. So again, a couple of predictions for this chapter. Essentially, we predicted that antler characteristics were going to be highly heritable because they influence reproduction. And we thought that by accounting for these different maternal factors of litter size, parturition date, accounting for those factors was going to improve our ability to predict antler size later in life based off of their yearling antler characteristics. So we used a full animal model, again, an MCMC GLMM framework to assess heritability. We had our variable, which essentially we're looking at 6 different characteristics: number of points, antler score, antler mass, inside spread, mean main beam length, and mean basal circumference. So those were our response variables. Then we included soil region, generation, and age in the model to account for that variation. And we included individual IDs or random effect to account for repeated measurements of those individuals across time. For our predictability analysis, we used the generalized linear mixed model, the LME4 framework, and program R. And again, we wanted to look at our ability to predict future antler size off of yearling antler characteristics. So our model framework was basically the relationship between 1.5 and 2.5-year-old antler size, 2.5, 3.5-year-old antler size as well. We included litter size and parturition date as a random effects, and then we calculated a marginal and conditional R squared. Essentially, a marginal R squared is the R squared of the fixed effects only, and the conditional R squared is the R squared of the fixed and random effects. So it allows us to basically assign a fit of those random effects. So we find that antler characteristics were highly heritable or lowly heritable. Well, our results show that all characteristics were moderately to highly heritable. But our heritability estimates weren't quite as high as Williams et al. Had reported, but they weren't quite as low as some of the characteristics that Lukafar and Jacobson reported either. Moving on to predictability. Well, these are the 3 relationships that we wanted to look at. So the conditional R squared of simply using 1.5-year-old antler size to predict 2.5-year-old antler size resulted in an R squared of about 45% of the variation being explained by yearling antler characteristics. That same relationship between 1.5 and 3.5 years of age, R squared was reduced a little bit to about 38% of the variation explained, but still a moderate amount of variation for biological standards. Within the relationship between 2.5 and 3.5, the R squared increased about 68%. So your ability to predict 3.5-year-old antler score dramatically increases when you let an animal get to 2.5 years of age. But when you include those maternal factors of parturition date and litter size, the predictability dramatically increases. For the relationship between 1.5 and 2.5-year-old antler size, you see a conditional R squared of about 85%, which means that those maternal factors are accounting for about 40% of additional variation in the model. Same story when you're looking at a relationship between 1.5 and 3.5-year-old antler size. Predictability nearly doubles. You have an increased ability of about 36% of variation explained that is accounted for by the maternal factors. But when you look at the relationship between 2.5 and 3.5-year-old antler size, the maternal factors had significantly less influence, with the predictability only increasing about 16% when you account for those maternal factors. So we know those maternal factors beyond a doubt influence yearling antler size because of the amount of variation that was explained additional to just our fixed effects. But that generally decreases once deer get older in life, as indicated by the relatively smaller amount of variation explained once we got to the 2.5-year-old age class. So once it's all said and done, you're trying to predict 3.5-year-old antler size based off of yearling antler characteristics. Counting for litter size and parturition date basically allows you a 95% improvement in predictability, which obviously is a pretty big deal. But if you're a manager, you don't know what parturition date and litter size is, so that's pretty important to understand. So moving on to the discussion, we see that secondary sexual characteristics are highly heritable for several different species. Antler and horn size, spotting variation in fish, feather coloration in birds. These are all highly heritable characteristics. But we still saw basically 3 studies with 3 different estimates of heritability for antler characteristics of white-tailed deer. Williams et al. Kind of represents one extreme, Luke Farr and Jacobson represent the other extreme, and our study was right in the middle. But there's a lot of differences among these 3 studies that could potentially explain differences in estimates. Heritability is a population metric, meaning that it's unique to that population. So if you have differences within a population or differences across populations, you have to understand what those differences are to compare those heritability estimates. One of the big differences between our study and Luke Farr and Jacobson's was regional variation. All of our deer originated from the state of Mississippi, and we accounted for that variation in our models. Luke Farr and Jacobson had deer originating from Ohio, Michigan, Virginia, Texas, Mississippi, all across the U.S., but they didn't account for any of that variation in their model structure. And all 3 of our studies had different age structure for our populations. Williams et al. Looked at heritability of yearling antler characteristics only. Luke Farr and Jacobson had a relatively larger sample size, so they broke it down and estimated heritability for yearlings, 2-year-olds, and then combined 3.5-year-old plus for heritability metrics of adults. And we assessed heritability of 2.5 to 5.5-year-olds. So we all had different age structures, which could potentially influence the heritability estimates that we derived. With predictability, we know that yearling antler size is influenced by those maternal factors of parturition date and litter size. We saw that by the dramatic increase in variation explained once you accounted for those factors. So they greatly influence those yearling relationships, but that influence decreased with increasing age. Again, when we looked at the 2.5 to 3.5-year relationship, maternal factors explain significantly less variation in those models. So that all leads us to suggest that maternal or antler characteristics can compensate if you allow these individuals to become mature. If you allow them to progress throughout time, some of these influences that they experienced earlier in life diminish. So bringing this back around to our question at the beginning of this chapter, can harvest alter the evolutionary trajectory of antler size for white-tailed deer? It's not likely. We're really inefficient at selecting and making predictions from those early age classes, and that's really where that question comes into play. But not only that, the breeding strategy of white-tailed deer doesn't allow them really to have this occur either. White-tailed deer display a tending bond breeding strategy. Essentially, one buck is following one doe during the entirety of their estrus, which occurs about 24 to 36 hours. And in northern populations where you have really condensed breeding seasons, that really limits the ability of a single buck to reproduce and have a lot of potential reproductive encounters. That's different from like elk or red deer where they have a harem breeding strategy, or even for bighorn sheep where you have increased, or age really plays an influence in that reproductive strategy as well. In whitetails, all age classes breed. So just because you might have an older age structure herd, yes, those older age bucks are going to do a majority of the breeding, but yearlings and 2-year-olds are still going to get in the breeding game as well. So that diminishes our ability to manipulate the genetics. And multiple paternity. About 25% of twin litters that are born from whitetail mothers have different fathers. So again, that just disperses the genetics across the population as well. So it's really unlikely that hunting is going to alter the evolutionary trajectory for several different reasons. So wrapping it all up, chapter 1, again, we saw that 3 populations of deer can switch from efficiency to luxury phenotypes once you improve their nutritional quality. And that antlers, the weaponry, is going to be respond the most to differences in nutrition. In chapter 2, we saw that individuals can display alternative strategies to potentially influence their reproductive attempts later in life. In chapter 3, we know that antler size and mass is highly heritable, but hunting is not likely to alter the evolutionary trajectory of antler size because of several different variables. So that, thank all of our people that we had working on this project. It was a 10-year project, so a lot of technicians, a lot of other graduate students. We had funding from Mississippi Department of Wildlife, Fisheries, and Parks, using funds through the Federal Lake and Wildlife Restoration Act. And with that, I'll take any questions.
Okay, so folks, so we'll take questions from the audience here for a couple of minutes, and then we'll give the folks in West River an opportunity to ask questions.
So the environment in which heritability is measured can also often influence the heritability estimates. And I was just curious about the actual environments. Were they captive, wild, in the 3 studies where you're trying to, to give it more context?
Yep, so all 3 of those populations were captive populations, all high quality nutrition, so similar rearing environments.
Okay.
And can you please repeat the question so that the remote people can hear?
Should we do that one too?
I think we can.
Okay. John?
On your second chapter there, you stated that lakewalk fawns can catch up then. Do you know how, did you determine how they're catching up? Is it protein or muscle mass? Or is it just additional fat stores in order to allow them to get bigger through time?
Okay. So the question was, did we actually quantify how late-born individuals in our second chapter increased in displayed compensation? Did they put on more fat or they increased muscle mass? We weren't able to quantify that. We just had body masses. So we don't know what the proportion was, if they were just putting on more fat or if they're putting on more mass across time. We did actually just do a simple histogram of different growth rates, and late-born individuals had increased growth rates across time as well. We just didn't know what they were investing in, either fat resources or muscle.
Have you related any of this to the literature of domesticated animals? Because it seems to me all of these things are really figured out. Obviously not the antler thing, but most of all these other things, because whether it's cattle or sheep or something like that, they have EPDs, all this from the genetics, how all this works out. Also, our colleagues in animal science have been doing some fetal programming things, kind of what you were talking about as well. And I just wondered, like, how does this relate to that kind of literature?
You mean just all this in general, or specifically?
Yeah, all of this in
heritability?
general, like, from the age, those kind of things, and the other related to John's question, there was another study where they took younger animals and got them at a different plane of nutrition, and later on looked at marbling in cattle.
Mhm.
And obviously what happened is those that are shorted, they aren't gonna marble because that's the expensive part, is the luxury part is the fat. So they're doing those, beating those other things first, and so that whatever happened early did have effect. So I suspect that perhaps when they compensate, they'll probably have to do what they need to be first, which is organs and then muscle and then
Sure, that clear
fat still
prioritization.
will happen later.
Yeah, so the question was, was I able to relate this back to the animal production side of things? With heritability, so we had an actual animal production scientist on our committee, and so he helped out a lot with that heritability stuff. I wouldn't be able to do that without him. And relating that to livestock, obviously livestock's a lot more controlled situation, a lot larger sample sizes. We were really restricted in our heritability analysis by sample size and age structure, and just a lack of information on heritability of antler size. Like I said, all these different studies, whether it's captive, you also have a lot of free-range studies that make the comparison even more difficult. So we did take that into consideration, and even the heritabilities are reporting are still much higher than what you see in with livestock. I guess the second question was with regards to the compensation that we saw in the second chapter. So yes, I mean, there's plenty of feed restriction studies that show that once you level the nutritional playing field, restricted animals can't compensate. They invest different resources or different organs first. But food was not limiting in our study. We were specifically looking at late born versus early born, all animals being fed on the same having the same nutritional field. So that was something that was unique about our study, was we weren't worried about nutrition. Nutrition was consistent for everybody all the time. We were just simply looking at late born individuals and how they responded to their environment. So, but yeah, I would imagine there's going to be some type of hierarchy even with that. There's going to invest in certain.
I wonder if ultrasounding would allow you to look at muscle versus fat.
It might. That'd be something to look into. Yeah.
Do we have any questions from West River folks or remote folks? You can go ahead and use your microphones and just shout it out. Okay, we'll take another question here.
John.
For your first chapter there, where you talked about I think it was the coastal plain deer that actually showed more of a significant increase in characteristics than, so moving more to a lecturing phenotype. Do you know what the mechanism was? I mean, it's been hypothesized that cortisol or changes in cortisol levels might be involved in that. Did you actually look at any of that?
So again, the question was, did we quantify any I guess, the mechanism that allowed our LCP bucks in the first chapter to compensate and have a more dramatic increase in their body size? We didn't. We weren't able to do that. One thing I will note, yes, LCP bucks did generally display the largest increases from first to second generation bucks, but those delta bucks, and even thin bucks to a certain extent, still had dramatic increases. Deltas particularly with their body weights. So we originally hypothesized that because those lower coastal plain deer in the wild, they're more nutritionally restricted. There's not a lot of high quality browse in the southeastern part of the state. We had expected that those individuals would have the greatest increases, but we saw really large increases for all three soil regions, which suggests that we can't blame it solely on the poor nutrition that is being experienced by those animals in the lower coastal plain region.
Is that lower coastal plain, is that longleaf pine, sandy
Yep.
soil,
Yep.
low quality?
Really poor quality soils, a lot of leaching problems. Primary land use in that part of the state of Mississippi is going to be pine plantations and cattle grazing, either of which really promote high quality deer browse production. So very, very limited in that part of the state.
So when looking at the epigenetics, you would think you'd need like a paternal half-sib design, right? You'd need a mom mated with other males to look at the progeny. And did you have those kind of data? I'm just curious if you had a few and what were the indications, or that's how you would really be able to show these cross-generational epigenetic
Sure.
effects.
So the question is the type of data that we had, whether or not we'd actually be able to parse out epigenetic effect of that first chapter. So generally, mothers were bred by different fathers across time. So we have a lot of half-sibs, but we didn't specifically look at that. We thought that quantifying the epigenetic effect was going to actually take more of a genetics type of analysis, where we're going to have to extract DNA and look at these different genotypes across time. So we didn't look at that. We didn't have, I guess, the technology or the ability to do that. So no, we were unable to actually quantify that epigenetic effect. We just hypothesized it because we saw that transgenerational response.
Any other questions? Any other questions from remote folks? All right, let's thank Eric one more time. And remember, next week, Pete Bauman and the tutorial on his data layer following that. Thanks, everyone.